Assalamu alaikum wa rahmatullahi wa barakatuh. Good day, everyone. We're still here with the National Science and Mathematics Olympiad, NSMO, the program that designed to build deep understanding and scientific skills and just build for you, yes, you, talented student. In this moment, we are in phase one of biology, where we focus on core and essential concepts. Today's lectures is a crucial part of understanding the cell and its vital functions. Hello again, this is me, Muhammad Al Ali. I'm your instructor in biology, and we're still here in chapter 1, and we discussed before cell structure, functions, and differences between cell types, and we also covered membrane components and the different types of transport across it. Today lessons completes the last major sections in this chapter. As shown in the outline, we have completed most chapter topics except membrane transport. This gives us the chance to understand how substance move in and out of the cell. We approach lecture 6 and it's titled Transport of Substances Across the Membrane. And in this lecture, we will learn how the cell controls its internal environment to maintain life. And before starting, let's recall what we learned previously. We studied cell structure, components, the cell membrane, simple division, and facilitated division. These concepts are essential for understanding today's lecture, so don't forget it. Okay, today we'll focus on four main transport processes. The first one, osmosis. The other one, active transport. The third one, endocytosis. And the last one is exocytosis. And all of these are essential for the cell survival and continuous function. The first main transport process we have it is osmosis. Osmosis is the movement of water from a lower solute concentration to a higher solute concentration. Look to this diagram, guys. Here shows water moving from the low concentration to high concentration. So what's the benefit for it? The benefit is help the cells maintain water balance inside the environment. And solutions are classified according to their osmotic concentration into three categories. Hypertonic, well, it is high solute concentration. Hypotonic, it's low solute concentration. Isotonic solution, it's equal solute concentration. The most obvious examples on osmosis is red blood cells and plant cells. And look to this pictures, guys, here. And this picture shows what happens to cells when placed in solutions of different concentration. The first scenario here, if we put it in hypotonic solution, the cell swallows here and may burst in red blood cells. And here in plant cells, it will be normal Trugel cells. The second scenario is in hypertonic solution. The cell loses water and shrinks. And here in the plant cells, the cytoplasm shrinks from cell wall. Okay, and the normal case in isotonic solution. In isotonic solution, the cell stays normal and balanced in both red blood cells and plant cells. Moving to the second main transport process, which is active transport. The active transport is the movement of substance from low concentration to high concentration using energy. Look to this picture, guys. This picture shows a transport protein using ATP energy to move ions across the membrane. And listen careful to this clip, guys.
Sodium-potassium ATPase, also known as the sodium-potassium pump, is a membrane-bound transport protein. It's an ion pump which functions by working against a concentration gradient and requires a direct energy source, ATP. This gives rise to the alternate name for this type of transport, primary active transport. Sodium-potassium ATPase has an intracellular ATP binding site. If ATP binds here, the protein's conformation opens up to the cytosol, eventually taking in 3 sodium ions. The protein changes its conformation and closes at the intracellular side. Now, autophosphorylation occurs, which releases ADP while phosphate still remains bound to the protein. Next, the protein opens towards the extracellular side, releasing 3 sodium ions into the extracellular space. In turn, the binding pocket becomes available again, and 2 extracellular potassium ions can bind. A new conformational change of the protein causes the binding pocket to close again. The intracellularly bound phosphate is cleaved hydrolytically, that is, by a water molecule. Through the subsequent binding of another ATP molecule, a new conformational change of the ion pump occurs that opens it to the intracellular side. The potassium ions are released into the cytosol. 3 sodium ions now bind to the empty protein for transport into the extracellular space, starting a new cycle. Sodium-potassium ATPase is one of the most important transporters for maintaining the vital concentration gradients of sodium and potassium. This concentration gradient, generated by ATP consumption, is fundamental for regulating water content in cells and electrochemical cell communication. It can also drive secondary active transport processes.
And be attention, guys. This type, which is active transport, is crucial for maintaining electrical gradients in nerve and muscle cells. So it's very important. The third process we have endocytosis. Endocytosis is the process that allows the cell to take large materials by folding its membrane inward. The membrane floats around the materials and forms a vessels inside the cytoplasm. And there are 2 main types. The first one, we called it phagocytosis or cell eating. So occurs when the cell engulfs solid particles like bacteria or cell diapers. Look to the right picture on the above. It shows the cell forming here, membrane extension to engulf this kind of bacteria. Okay. The second part of endocytosis, it's pinocytosis, or they called it cell drinking. And the cell drinking, it's the cell takes in, dissolves solutes and extracellular fluid into small vessels. Look to the pictures below, down here. We see the lower image here illustrates dissolved solutes being internalized in tiny vessels. This is the tiny vessels that we talking about. So this define what we know as endocytosis. We reach to the final transport process. We called it exocytosis. Exocytosis is the opposite of endocytosis. The cell uses it to expel materials outside. Vessels move toward the membrane, fuse with it, and release their contents outside. Look to this picture, guys. In this pictures here, let me clarify it. Here, the image shows vessels containing neurotransmitters fusing, releasing their contents, like this one here. Okay? Let's have an exercise. So, a cell is placed in a solution with a lower solute concentration than its cytoplasm. At the same time, it needs to import large lipid tubules and export neurotransmitters while maintaining its internal iron gradient. Which combination of processes will the cell most likely for the cell use to accomplish all these tasks? Is it osmosis for water entirely, active transport for ion pumping, endocytosis for lipid uptake, and exocytosis for neurotransmitter release, or active transport for water, interosmosis for ion pumping, endocytosis for neurotransmitter release, and exocytosis for lipid uptake? C. Exocytosis for water entry, active transport for lipid uptake, osmosis for neurotransmitter release, and endocytosis for ion pumping. Or D. Osmosis for neurotransmitters release, endocytosis for water entry, exocytosis for ion pumping, and active transport for lipid uptake. Think about it, guys. What do you think? Let's analyze this. Water enters via osmosis, okay? Ions require active transport to maintain gradients. Large liquid droplets enter via endocytosis. Neurotransmitters exit via exocytosis. So, for sure, the answer would be A. Yes, you are right. Why A? Because A contains all correct combinations. What is all the correct combinations? Osmosis plus active transport plus endocytosis plus exocytosis. And A is the only option that correctly combines all these main transport processes. So the answer would be A. Before we reach to the end, we covered in this lecture osmosis, active transport, endocytosis, and exocytosis, and we know the differentiation between them. Thank you, talented students, to be with us. Thank you so much. See you soon. Stay tuned. See ya.